Okay, let's go over one of the most key skills in microbiology, which is microscopy. So yeah, general overview of microscopy. I'm sure all of you guys have done it, in science class, and it's an essential tool for researchers because it allows us to visualize individual cells, materials, particles, and actually you can even see individual atoms nowadays, which is like really insane. So if you're curious about that, you can ask me about it sometime, but I'm not going to include it in this lecture here. All right, so just brief history. These are some pretty important guys that you should know of. Antony van Leeuwenhoek, I think that's how you say his name. This guy, we call him the father of microbiology. Basically, in the 1600s, he got a simple microscope, so a single lens microscope, almost like you can think of it like a magnifying glass, right? And then he started looking at like pond water, and then he saw all these microbes. He nicknamed them animalcules. And then he documented all the microbes that he found, and he was kind of the first person to start doing this. And then he eventually he wrote a series of like reports and documents that got sent off to like I think the like English Royal Academy or the whatever the institution was then it got sent off there. So and then we have this other guy Robert Hooke. He did the similar thing too. He also drew diagrams of all the organisms that he saw in this pond water, and he used a compound microscope, which has like two lenses. So I think the important thing here is the term. Simple microscope is one lens. Compound microscope has two lenses, kind of like a telescope, right? All right, so let's start with the bright field microscope, or you can call it the light microscope, right? These are the most common type of microscope. It's like the ones that you have been using in your entire scientific career so far, right? So they have relatively low resolution, and resolution just refers to how clear the image is, and we'll get into a little more specific definition in a few slides, but for now you can think of it like that. And it also has pretty low contrast, so there's not stark like darks and lights, right? It's pretty kind of just darker. And the image itself is darker than the background. The background is usually like white or gray, light gray, something like that. So these are particularly useful for when we have, where you can kind of use them with anything. Live samples, fixed means like killed samples, dead samples. Stained and unstained. Stained, you put some dye in it so that the color is more, there's more like contrast in the image with the dye, and then unstained would just be like the cells themselves. But the staining cells is like highly preferred because it allows you to specifically visualize structures that you want to see and cells that you want to see. So here, the max magnification is usually around 1,000 times magnification, and at this magnification you can actually see bacterial cells. So you can see within the micrometer range, these. They'll probably quiz you on the different parts of a microscope. So I would either put some diagram like this on your cheat sheet, or the safe way to do it would just be to go through the Quizlet that's on the Google Classroom that I made last year, and then just like memorize based off of that. It's not too bad. But basically, right, you have the ocular lens. This is the thing you look through. Then you have the body tube where the light is traveling to your eye. You have the nose piece that can rotate around, and then which is the objective lens. Some of them have higher magnification than others, so you can rotate that nose piece around. And then, of course, you have the stage. You place your sample. You have stage clips to help keep your sample on the stage. Then you have this diaphragm. So that diaphragm is just going to control the amount of light that gets into your sample. Then you have the light source here. So you can open and close that diaphragm. And there's also, you can think of this also called a condenser. And technically these are two separate things, but you can kind of think of them the same way. So you can like change the amount of light that's getting let in by opening and closing. Then of course you have the coarse adjustment, which is like the big knob that, basically it's not fine tuning, it's like kind of big adjustments when you're looking at it under the microscope. And then you have the fine adjustment, which is like fine tuning. You can slowly turn that little knob and then you'll get a little better image or a sharper image. And so you'd always start by using the coarse adjustment, then using the fine adjustment. Okay. So yeah, here's some just images from bright field microscope, pretty standard. We'll talk about this stain in the next lecture, but you can see these bacteria here. Then on the right, this is actually a sample from like Cassidy that I took a picture of this in Mr. Del Grosso's room. So it's just a bunch of like algae and what we call diatoms. These are just like floating around in pond water. So pretty cool structures there. Yeah, so you can also have a light microscope or a bright field microscope that's inverted. So it's the same exact mechanism, but this time the light source, usually you have the thing like this, and then you have the light source from the bottom. This time the light source actually comes from the top. And then the objective lens, right, where these like lenses here are actually on the bottom here. So what that does is that our view is from the bottom like this. So we see, if we have like a slide here, we're going to see the bottom of the cell, or the bottom of the slide, or the bottom of the container that contains our specimen. So this one's especially useful for like, let's say you're culturing, so you're growing a bunch of like, I don't know, like endothelial cells. You're growing a bunch of them in this like container, right? Let's say you have like a flask like this. So you have some media, some like water stuff, and you have all the cells here, right? All these cells, since they're denser than the thing that they're floating around in, right? They're going to float to the bottom. They're going to stick to the bottom here. So for that scenario, we'd use an inverted microscope, because then we can see, our vision will be from the bottom, so that we can specifically look at all these cells, as you can see here. So yeah, live, often used for live liquid samples where the cells will stick to the bottom, and cell culture. Yeah, cell culture is just growing cells in a lab in one of these like boxes here. Yep. So basics of microscopy, right? The light comes out of the illuminator. This bottom part here, this is not inverted, right? Then it passes through the specimen. As it goes here, it then passes through the objective lens. This first lens here keeps going through the body tube, goes through the ocular lens, then finally it'll reach your eye here, like this. So that's how the light will travel for the majority of the microscopes that we'll see here. All right, so let's talk about magnification. So you can calculate the total magnification that your microscope has, and you'll do that by multiplying the magnification of the ocular lens and the objective lens. So here, right, you have the objective here, the ocular lens here. So if this is like, let's say this is 10 times, and then this is 10 times, your total magnification would be 10 times 10, 100 times. So you can see that here. And then, for example, ocular is 10, objective is 100, total magnification is 1,000. And remember, this is the max, usually for light microscopes. So for the ocular lens, the one that's close to your eye, the magnification is almost always going to be 10 times. That's just like a standard practice that we do. And then usually you will have 3 or 4 options for your objective lens. And this is like, remember this, where is it? Right, this rotating part here, like we have 3 different objective lenses here, right, at different magnifications. And for the vast majority, these will be 4x, 10x, 40x, and then if you have a better microscope, it'll go up to 100x. And then we have names, which I would know for each of these magnifications. So 4x is called scanning power or scanning objective. 10x is low power, 40x is high power, and then 100x is what we call oil immersion. And we'll see exactly what that is in a second. Yeah, so key tips for when we're using bright field microscopes. First of all, only use the fine adjustment when you're at these 4x or 10x, these lower powers, okay? Because as we increase the magnification, right, so if this is the slide here, and then this is the objective lens, right, sticking out, looking towards the slide, as you increase the magnification of the objective, this thing's going to get longer and longer, and it's going to get really close to the slide here. So you don't want to use the coarse adjustment, right? The coarse adjustment is the big knob that that if you turn, it moves the thing a lot, right? The microscope. You don't want to use this when you're at high power or oil immersion or 100x, because the objective lens is so close to the sample that if you turn it with a coarse adjustment a lot, then the lens can just like break through the break through the slide that we're looking at, and then it'll just like, that's like really bad because you don't want like glass shards just like flying everywhere. So make sure you don't use the coarse adjustment when you're at higher powers. And then always use the oil with the 100x. So we'll see what this means here. Okay. And another thing to know is that you're always seeing the cells or the image in what's a backward mirror image. So if the cell looks like an E like this, then you're always going to see the exact reverse like that. So what you're seeing is like the opposite pretty much. All right, and then just some general, I would probably, I have this on my cheat sheet, so but I would just know this. So when we're increasing the magnification, right, the FOV, like the field of view, how much we're seeing within the like field of vision in that circle, it's going to get smaller and smaller, right, because we're zooming in more, so we're seeing less of the sample. And it's also going to get darker. I don't exactly know the reason why, but you'll notice that when you use the light microscopes like at school, if you go up to like the highest magnification, the sample will get really dark and like kind of grimy. So then the resolution decreases because we're getting closer and closer. You get less, you get less detail. It's like if you're looking at your computer and it looks normal, but you put your eye like straight up to the computer and you see all the pixels, right? So it's not like you don't see the image anymore. It's kind of that idea. The working distance decreases, so the distance from the objective lens to the specimen here is going to decrease. Then what we call the depth of focus. I don't actually really know what this means, but I read it, and I think it might show up, so I would just write this down somewhere. But honestly, I am not exactly sure what this means. Okay, so oil immersion, right? We talked about how when you use 100x, the highest magnification, you need to use what's called oil. So what we're going to do is between, like as you can see here on the right here, between the objective lens and the slide, we're going to put a little thin layer of oil right here. We're just going to pour some oil on top of the sample. Basically what this does is, so I'll start by explaining the physics a little bit. So when you have light come out of the illuminator here, the light source, right? Air is very not dense, right? So the light just travels pretty straight through air. But when it hits this glass slide here, the glass, right, is a lot denser than air. So what happens is that the light kind of warps and it gets bent like away from the sample or from the objective lens. But we want all the light to go through here so it can see the sample as best as possible, right? So basically at high magnification, so when we're at like 4x, 10x low magnification, it doesn't really matter because the objective is like way up here anyways. It's pretty far away. So like, we're just doing the best we can. We're not collecting all the light, but like it's okay because we're not zooming in super far. But when you're at 100x or, this is technically 1000x, because you're multiplying the ocular lens times the objective lens here. So technically you're looking at 1000x. You want the resolution, right? You want to collect as much light as possible because you're looking very zoomed in. And if you lose a little bit of light, you're going to lose all that detail really fast. So the oil, it has the same what's called refractive index as the glass. So basically the light, it'll bend as it's going through the glass. So if this is the glass, it'll bend, but then there's oil here that kind of traps the light in and then keeps it moving straight. So you can see that on the right here, right? So that oil, it allows us to collect all the light that's coming through the specimen so that we can see the best image possible, essentially. All right, so let's briefly talk about what resolution exactly means. So resolution, we can think of it as the ability to distinguish two nearby objects. So on the right, let's say so you have these like two green particles, right? So if you look under a microscope and you see this, that means that, the resolution is pretty good because you can see both particles as distinct objects, right? But then as we move them closer and closer, you can see that under the microscope, it'll just look like one blob. So even though there's two distinct objects, the resolution of the microscope is not good enough to see them as separate objects, so it just kind of blurs into one. So that's what resolution is saying. It's like, so basically the ability to distinguish two objects that are really close together. So very high resolution means that even objects that are really tiny and close together, you can still tell them apart from the microscope. Yeah, so the reason, like, in a cell, you'll have proteins and like mRNA and all this crap, right? Like all this crap just floating around everywhere all the time. But like when you look at a cell under a microscope, like you're never going to see the proteins like floating around, right? So, and that's the reason why, because first of all, they're flying around insanely fast. And second of all, the resolution of the microscope isn't good enough to distinguish those flying around tiny proteins as like individual objects. So it's just kind of a blur. Everything just looks like the cytoplasm. So we can actually mathematically determine the resolution of a microscope, and I actually didn't see this show up on any of the tests that I took last year, but I'm gonna cover it just in case. So basically, we have this value called aperture, and think of it like the ability of a lens to gather light into it, and then light coming around. So a higher numerical aperture will mean that a lens is better at collecting a lot of light, right? And then basically, like if you have a high aperture, that means that you collect a lot of light, and that'll make your image a lot better, right? Okay, so what we can do is we can relate this aperture to resolution by using what we call the Abbe equation here. That's this thing on the right. Here, D refers to, you can think of it as the resolution. And then this NA is the aperture. Or we call it the numerical aperture, same thing. And then lambda, this is a Greek letter called lambda. If you guys have taken physics, you'll know. But if not, that's totally fine. So lambda refers to the, this is very scuffed, wavelength of light. So the wavelength of the light that's coming out of the light source that you're using for your microscope. So you can relate this in this equation here. So if you have like two of the values, you can determine the other one through this formula. So when D is very small, that's what we want, because the resolution, the D is referring to how close we can, it's determining like how well we can look at objects that are close together. So if the D is small, it means we can see objects that are closer together better, essentially. So small d value corresponds to better resolution. And if we want to make d smaller, right, we can increase the denominator. We can make the numerical aperture larger so that we collect more light, and then that'll make our resolution better. Or we can make the lambda, which is the wavelength of the light we use, very small. So we can use higher energy light that has a smaller wavelength in order to increase the resolution. And we'll see this in future slides, because for like normal light microscopes, we're just using like standard visible light, right? But then later on, we can actually use like electrons instead of light. And electrons have really, really small wavelength. So that'll correspond to a very high resolution, which means that when we use electrons to look at a sample, we can zoom in really, really far and see tiny, tiny things a lot better. Hopefully that made sense. If not, let me know and I can try to re-explain it live. Okay, so and then we can also calculate the field of vision, so how wide the image is that we're looking at. And the easiest way that they'll usually make you do is they'll just give you a clear ruler that has tick marks on it at 1 millimeter, and then what you do is you literally just place the ruler under your microscope. You look at it under the microscope. Then you see how many tick marks wide the field of vision is, and then you can determine how wide the field of view is. So that's not bad here. That's pretty much all you have to do. Okay, so dark field microscopes. These guys, they show so the image itself is light, but the background is like black. It's very dark. And dark field images, they don't require staining. And I feel like the important thing here is not necessarily understanding how all these microscopes work, but more so what they're good for, like when they should be used, and kind of what advantages they have and disadvantages. So here, the advantage of dark field is that your samples don't require staining. So we don't have to put dyes onto the cells to be able to see it well. And that's good because you can look at cells, because some cells, when you add like dye to them, they're gonna like get damaged or die. So if you use dark field, then you don't have to stain those cells. So those you can just look at them when they're alive instead of killing them with the dye, essentially. Yeah, so this will give you a higher contrast than bright field because it's like a lot darker and you can see the light portions better, and it has about the same amount of magnification here. So essentially, this, I don't think it's that important, so I think I will skip this for now. But essentially, you're letting a lot less light, you're letting a lot less light into, out, or like a lot less light out of the light source, and then it makes the image very dark. And we'll get these very colorful, vibrant, pretty high contrast images, as you can see on the right here. Pretty cool. Okay, phase contrast. These guys, we get a high contrast image against a gray background. And again, you don't have to stain these because basically the microscope will enhance the contrast for you. So you don't have to increase contrast by like staining the cells. And it requires this thin sample. Yeah, so this is some physics stuff that I was wondering if I should explain or not. Basically, I think it's okay. Just know that the phase contrast, essentially, if you just look at a cell, it doesn't have that much contrast because it's all clear. But the phase contrast will make it so that the clear cell will have more differences in like darkness and lightness, essentially. So that's how you can think of it. And then they also create this halo effect around the image. So if you look on the right here, you see you have a gray background, you have these like highly contrasted, almost like black and white type of image. Then you have this halo, this white halo light area around each cell. You have that same thing on the right here. Okay, differential interface contrast, DIC. So here's a case where we're using infrared light. So infrared is like, think of like heat vision, right? So that's the kind of light we're using, and that allows us to get a little more high contrast resolution. And we can also use live unstained samples here like we did in the dark field and in the phase contrast. So yeah, the image is pretty sometimes colorful, and it looks, you have this like 3D effect. And you get that effect because basically what you're doing is you're using what's called a Wollaston prism. So when this light comes out, you kind of use the prism to like split the light into these two paths like this. So basically it's kind of like you can see different angles of the sample because you're like splitting the light this way. I mean, that's kind of how you can think of it, I guess. And then can be used for thicker specimens. Yeah. And then we don't have the halo effect here. So you can see you get that kind of 3D effect, right, where you can see things protruding and sunken into each of the cells. And again, it's very, first of all, these microscope mechanisms are very complicated, like physics-related things. So one is that I don't fully feel comfortable in like my ability to explain these things. And second is that they're not really relevant for the purposes that we're looking at, which is why I'm kind of glossing over all of these. But like I said, the important thing is to just know when what the advantages and disadvantages are for each microscope. Then if you look on the cheat sheet that I made, it'll have all of these down, and it'll tell you like what's good, what's bad, etc. So you just refer to that if you need. All right, so this one's pretty significant. So fluorescence microscope. Basically in this one, what we're going to do is we're going to stain our sample. So we're going to add a bunch of dyes to our sample, and these are called fluorochromes. And these are like bright dyes that glow in the dark. So if we put these dyes that bind to specific locations in the cells, if we look at them under this microscope, then we can see them like specific things glow in the dark. So for example, let's say we use a dye that binds to DNA, like a blue dye. Then we look at it under this microscope, and then when we see the cells, we'll see all the nuclei, but like glowing blue, so we can see exactly where the nuclei are. It's a pretty useful technique. And we can also use what are called fluorescent antibodies. Antibodies are things that bind to specific proteins. So, for example, let's say I want to see like, what's a useful protein? Like I want to see actin in the cell, right? Part of the cytoskeleton, right? All those like little line-like things that like stick off everywhere and like give the cell structure. Then I could insert an antibody that binds specifically to actin, and then all the actin molecules will start glowing, pretty much. Yeah. So yeah. So again, this is a lot of physics related things. So okay, the thing is, you should probably know that a mercury arc lamp and dichroic mirror are used in fluorescence microscopy. If you're interested in this, I can specifically cover this in the future, but I don't think that it's fully necessary to go into the physics details here for now. Yeah, so they give these like really beautiful images. So you can see we have the blue nucleus stain, we have these green stains probably for actin or tubulin, or the microtubules, and then you can stain bigger structures too. So this is like a fly embryo. You can see all these different cells stained. Yeah. All right. So finally, electron microscopes. So like I said, these use electrons to visualize images that are at the nanometer scale. So 1,000 times, right? 1 nanometer is 1,000 times less than 1 micrometer. So you're zooming in a lot more, and that means that you can actually look at viruses using electron microscopes. Because if you remember, viruses are within the nanometer scale here. Yeah, so this TEM, transmission electron microscope, this guy, we can get 50 million times magnification, which is like absolutely insane. But what we have to do is we'll slice the specimen into these 20 to 100 nanometer thick slices. And what happens is that we shoot this guy, this like thing here, it's like an electron gun. Electron gun. So we'll literally shoot the electrons at our specimen. And if there's like, in a specimen, there's like dense and less dense regions like throughout the entire thing, right? So if the electrons pass through a less dense region, then if the electrons hit a less dense region, it's not that dense, so they can just pass through. So like the electrons will just pass through a less dense region. But if there's a dense region, then the electrons will like kind of get bounced off like this. So this whole system is like measuring how much electrons get bounced off and how much pass through. And it'll give you this 2D image that has like darker and lighter places here. So you can see on the right, or not on the right, like on the whole slide, I guess. But right, these are phages, right? If you remember, those phages that, had that icosahedral head with those legs like this. This is actually what they look like. Then on the right, you have some bacterial cells here. So these darker areas are where you had denser regions, and then the lighter areas are where there's like very less dense areas. Yes, okay. All right, and then we have scanning electron microscope, SEM. This one, again, we're using another electron beam, so another electron gun that we're like shooting at the specimen. But this time, we're measuring the emission of electrons from the specimen. So basically, when you shoot electrons at something, that thing itself will release electrons as well. Again, a lot of physics-related stuff. So, but essentially, you can measure how many electrons are being shot off of the specimen in response to the electron gun, and you can measure that. And then if there's more electron release, it means the area is like higher up. It's like elevated. There's like a bump like this. Then if there's less electron release, it means that the region's like sunken like this. And it gives you this 3D image because you're looking at the actual contour of the specimen. And then key bit of information that they might ask you about is that we need to cover the specimen in heavy metal film, like a gold plating, because this basically it increases the amount of electrons that are shot out of the specimen when we shoot the electron gun at it. So it kind of enhances our image. And these images are really, really cool. So, and you can even like search up more on Google, just like scroll through these. They're very insane. So like you can see all these bacteria. You can literally see the 3D structure of all of them as if they were massive and you were just looking at them with your eyes, right? You can see this phage here, right? Phage. Like that, right? Yeah, pretty awesome. And then you can see these phages actually like attacking and infecting these bacteria here live. So I think this is probably my favorite one. It's pretty insane. Then finally, if we're trying to estimate the size of different microbes based off of like a photo, this is not bad at all. So essentially, you'll be given an image like this where they give you a scale bar, right? The scale that tells you how long that line is. Then all you have to do is like, if this small thing is 30 micrometers, then you just see how many bars can I fit here, right? So let's say I fit like 5 bars. That means this thing is like approximately 150 micrometers. So that's like the easiest question they'll give you. So that hopefully should be just like a free question, right? And then you also might be asked to. So remember when we had to use the ruler here, right? Where is this? FOV. Yes, here. So remember we line up the ruler and then see how wide the field of vision is. So you might be asked to find how big like a microbe is that you're actually looking at through the microscope. So what you'll do is you'll measure the FOV diameter here using the ruler, and then you'll look at the cells. And let's say if you can fit 4 cells in this diameter, that means that each cell is just 1/4 of the FOV. So that's how you would calculate the size of a microbe based off of the ruler method here. Yes, and I know this lecture might have been a little bit confusing, so please feel free to reach out if you have any questions, and I'll try to answer them to the best of my ability. Hopefully this helped.